Let's get our Friday underway. Get our business done and get out of here and go out and freeze our tails off.
So cold my car wouldn't start this morning.
It was so cold that, all right, how many of those do we have today? It was so cold I literally froze as I was running this morning. So, okay. I'm going to finish up some general things about transcription and processes related to transcription. And these will include some kind of unusual exotic modifications that occur to RNA molecules. So I want to get into that and say something about that. Last time when I finished up, I was talking about polyadenylation. And polyadenylation is, I will remind you, something that happens to eukaryotic messenger RNAs. It doesn't happen to other RNAs. It doesn't happen in prokaryotes. Okay? So it is a phenomenon unique to eukaryotic messenger RNAs. And it happens as a sequence AAU-AAA is encountered by the polymerase. The polymerase continues making RNA past that sequence, but an endonuclease comes along and clips this guy right here. All right? So that we see that a specific end is made to the RNA, even if the polymerase is continuing on. So we have a different termination mechanism here than we do in prokaryotic cells. A polymerase, as it's so called, then adds a sequence of A's near the 3' end. And the length of that sequence of A's, that is the number of A's in that sequence, is related to the stability of the messenger RNA. Longer means it stays around longer. More A's means it stays around longer. The reason for that is nucleases work from the 3' end inwards, and so they don't work very fast, and they start chewing through these A's. The more A's there are to chew through, the longer the A's are on there. Now, as we'll see when we talk about translation, just like the cap plays a role in translation, so too does the poly-A sequence. So if, for example, a nuclease chews all the way through the A's and there's no A's left, then this portion, which is part of the translational mechanism, isn't available. This messenger RNA isn't really going to be translated once those A's are gone. Okay, so somebody asked me last time about, well, what about some of these truncated proteins? Will they be a problem? And the answer is they're not made with any efficiency at all because of the lack of a poly-A sequence. Okay, so those are some modifications that happen to eukaryotic RNAs. I want to talk about a couple of specific things, including a process called RNA editing. All right? Now, RNA editing is primarily something that we see in eukaryotic cells. I don't know of editing mechanisms in prokaryotes, but there may be some. But editing is a much more common phenomenon in eukaryotes at a minimum. All right? Now, one of these things that I want to talk about is a very interesting edit that happens. So what is editing? Well, editing is a chemical modification that essentially changes a base after the RNA has been made. Okay, so it changes a base after the RNA has been made. A prime example of this is something that occurs in our intestinal cells. In the mucosa of our intestines, we have an enzyme that is capable of removing an amine group off of a specific cytosine in a specific messenger RNA that codes for a lipoprotein. All right? Now, if you remember, we talked about lipoprotein complexes and how they played a role in carrying fat and cholesterol and so forth across cells. It turns out that there is one that actually serves a dual purpose. All right? In intestinal mucosa, they have an enzyme that makes this modification. So there's a CAA that is converted to a UAA. That is, a C is converted to a U. A UAA, as we will see, is a stop codon, meaning that when the ribosome hits this, it stops translating. This means that in the intestines, a truncated, short version of this protein is made. It's only made up to this point. We call this protein ApoB48. It's just one of the proteins that's in the lipoprotein complex that comprises chylomicrons. Okay? Now, interestingly, in the liver does not have this enzyme with the ability to make that modification. So in the liver, what starts out as a CAA remains a CAA. CAA is not a stop codon. So read-through happens, and it reads all the way to a different stop codon considerably further away. In fact, about 2,400 nucleotides away. This makes a protein in the liver called ApoB100. And the 100 and the 48 refer to the sizes. That's 100 kilodaltons. This guy is 48 kilodaltons. So this guy is considerably bigger, and this guy is a component of LDLs. Now, that change of one base changed considerably the protein that was being made. Okay? And the only difference was liver cells don't make that enzyme, intestinal cells do. Now, there are other examples of editing that occur, and some of them are rather elaborate. One of the most unusual schemes for editing is actually found in a lower eukaryote known as the trypanosomes. And what trypanosomes do is kind of bizarre. They will make messenger RNAs, okay? And then they will, in fact, after the messenger RNA has been made, insert U's at various places in that sequence, okay? They're actually physically inserting, not replacing, not changing, but physically inserting U's into that messenger RNA that was made. That insertion of those U's is critical, because if they don't get in there, the protein coding is all screwed up. Now, it's a bizarre scheme. We refer to this process as editing also. It's a different kind of editing. We're not simply changing a base from one to another, but we're actually physically inserting bases in there. And this phenomenon happens primarily in trypanosomes, okay? Very low-end eukaryotes. Why it does it is anybody's guess. All right, so that's those two processes are examples of what I refer to as RNA editing. Another process that is found in eukaryotes but not found in prokaryotes, essentially, is a phenomenon known as splicing. Okay? Splicing involves chopping up a given RNA molecule into pieces. Some of those pieces are removed and do not appear in the final RNA. The ends of the other pieces are joined together. Now, your book talks about this happening as if it happens only in messenger RNAs, and that's not correct. Splicing happens in ribosomal RNAs, it happens in tRNAs, and it happens in messenger RNAs. The phenomenon is quite widespread. Most genes have, in eukaryotes, most genes are spliced. Okay? Now, splicing, as I said, involves basically removing a chunk of sequence and joining the other ends together. Okay? In the old days when we had reel-to-reel tape recorders, all right, if you wanted to edit sound, you had to physically cut out with a pair of scissors the piece of tape that you didn't want to have in the final product, and use a little adhesive to join the other two ends together to make what we call a spliced recording. Okay? And it's the same exact sort of phenomenon. We were removing a piece of tape in that case that had some audio that we didn't want, and we joined the ends like this. Now, I know I've just shown my age immensely with that, but that is an example of splicing in a real-world situation. That's also how they used to edit Hollywood movies, okay? Where you would physically cut the movie, take out a piece you didn't want, and join the other two ends together. I know it's the dark ages, but that's the way it was. Well, these guys have names. So the ends, the portions of the RNA that end up in the final product are called exons. E-X-O-N-S. The sequences or sections that get removed are called introns. Now, one question is, how does the cell know what to remove? We know a lot of the answers to that, but we don't know all of the answers to how the cell knows which sections to remove. You can imagine that if the cell doesn't remove a sequence properly, it's not going to make the right protein. Okay? Well, what we do know is the following. Okay? We do know from examining all of the introns that are known that essentially all of them have what's called a conserved sequence at the 5' end and a conserved sequence at the 3' end, and a conserved sequence in the middle. I'm going to describe these. When I say conserved, meaning that they are found across a wide variety of them. All right? At the 5' end, the only absolutely conserved sequence among introns is GU. GU must be kidding, right? GU is found in all of them. This is a consensus sequence where we'll see some of these other sequences appearing in some introns but not others. But GU is found at the 5' end of all introns in eukaryotes. At the 3' end, we see the sequence AG. And it's found at the 3' end of all eukaryotic introns. About 20 or 30 bases ahead of the AG, there is almost always a sequence that is very pyrimidine rich. Pyrimidines meaning C's and U's. And in the middle of that is an A. That A plays a role in the splicing mechanism. Okay? So 3 conserved features: a GU at the 5' end, an AG at the 3' end, a pyrimidine-rich region that has an A. Okay. When we look at what happens in the phenomenon of splicing, we see something like this. This isn't the best figure in the world, but let me try to explain what's here. Here is what we would call a pre-RNA. Pre meaning it hasn't been spliced yet. This pre-RNA has an exon. So our 5' end is up here. We loop all the way around to the 3' end over here. Okay? Here's the end of the exon. Here is our GU. Okay? Over here, we see our AG. All right? And here is this pyrimidine-rich region that has this A sequence. Everybody with me so far? The phenomenon of splicing involves a nucleophilic attack by this hydroxyl onto this phosphate over here between the GU and the AG. So these guys are brought into close proximity. There are some proteins, and I'll briefly tell you about those proteins that play a role in this process. Those proteins help to bring these guys into close proximity and allow this attack to occur and breakage of the bond between this guy and this guy. Okay? Everybody with me so far? This, as you see here, is labeled the branch site because, as we will see, this is making a branch in the intron. Now, before I tell you what happens up here, I want to tell you what we've got right here. What we have right here is something very unusual. In eukaryotes, we see some unusual bonds. Remember, we had the cap sequence, or we had the only place we saw a 5' to 5' linkage? Here is an example of an unusual linkage that we see, where we see a 2' to 5' phosphodiester bond that's been created. The result of this attack is that a phosphodiester bond is created between position 2 and that phosphate. Okay? Now, so at this point we have something that starts to look like what people describe as a lariat. This is the lasso part of the lariat. But we haven't finished the splicing process. To finish the splicing process, we have to attach the 3' end of the exon to the 5' end of the other exon. Now, this figure unfortunately shows, okay, well, I've got to turn this guy up. So if I take this guy here and I rotate it up, the 3' hydroxyl now attacks this phosphate between the 3' end of the intron and the 5' end of the exon. That breaks this bond, results in a spliced product. We've joined the 2 ends together, and we're left with what was the intron. The intron is now in the lariat structure. Now, the most common question I get is, well, what happens to that lariat structure? For the most part, nucleases break it down. For the most part, nucleases break it down. So this is one of the ways we've got to salvage nucleotides. When we break this down, we're going to make nucleoside monophosphates. In some cases, and these are unusual, in some cases, there are sequences in here that can be translated. Those are rare, but in some cases, sections in the excised intron can be translated. That's interesting, and we won't talk about those. But it says that cells use everything to their advantage. And in this case, rather than simply throw it away, in some cases it actually translates something there. All right, so this joins two exons together. Cells may have as many as 50 exons for a given messenger RNA. Most genes have on the order of, half a dozen. Not uncommon. Shannon, do you have a question?
Yeah, if that's translated, is this happening in the nucleus or in the cytoplasm?
Good question. Is this happening in the nucleus or the cytoplasm? This phenomenon of splicing is occurring in the nucleus, which means that this has got to get out to the cytoplasm if it's going to be translated. You bet. Okay, other questions about that? Introns are really interesting. They're unusual. When people first discovered them, they didn't think they were even possible, because why in the world would you want to interrupt a perfectly good coding sequence with basically what appears to be, and here goes this phrase again, junk DNA? Okay? And again, we don't have all the answers to it, but it appears that introns facilitate rapid evolution of proteins. Okay? They facilitate rapid evolution of proteins. How does that work? All right? When we look at exons, we look at the sequences that make it into exons, and now we're looking at messenger RNAs specifically. When we're looking at exons, and we look at the exons in messenger RNAs, we discover that roughly each exon corresponds to a specific protein domain. Now, what do I mean by a specific protein domain? Last term, we talked about domains very briefly, and we said they had specific structures, usually with specific functions. And a good example of a protein domain might be a region of a protein that binds ATP. A lot of cells bind ATP. Well, if I have an ATP binding region on a specific exon, it means I don't have to evolve that for every single protein. If I do mixing and matching of this exon with other coding sequences, now I can bring this domain into another protein and allow it to bind ATP and do something with it. Okay? To have the same thing happen in a prokaryotic system, we can still have domain swapping and so forth, but it has to be precise. It has to be exact. If I'm off by one nucleotide, what happens? I don't get a functional protein. Here, I can make the swap, and I can, over evolutionary time, swap sequences. And it doesn't matter if I do it here, 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 or here, or here, 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 or here. I'm not touching the exons. Okay? So the actual cutting and pasting of those domains is much easier to do if they're located on exons. Introns are variable. Exons, of course, have functional protein sequences when we're talking about messenger RNAs. I don't want to mess with those as much as I want to swap pieces. So I've basically got a little cutout area where it doesn't matter if I make a precise cut and swap that sequence with something else. That's one thought about the importance of introns. There are others. Okay, questions? Quiet group today. Okay, so that is the schematics of the splicing phenomenon. There is what that branch point looks like, and here's our 2' to 5' bond. Okay, so this guy would make a loop all the way around here like this. Okay. Notice that this one nucleotide that is at the junction of the lariat has both a 2' to 5' and a 3' to 5' bond. That's the only place in biology we see that, right here. Okay. Now, well, how is it that cells do that splicing? Does it magically occur? In some cases, it almost does magically occur. Okay? And it happens in most cases as a result of action of some proteins and some special RNAs. Okay? Those proteins have an abbreviation called SNRNPs. Okay? SNRNPs they have names, U1, U2, U5, U4, U6. Okay? These are proteins that make a complex that perform the splicing. Okay? They make a complex that form the splicing. They don't do it by themselves. They also do this in conjunction with some specialized RNAs known as snRNAs. So what they're making is a protein RNA complex, not unlike a ribosome complex, except for these guys are doing splicing, not translating messenger RNAs. Okay? So we have snRNPs, we have snRNAs. You can see the size of these. They're not very large. 165 nucleotides is the biggest one that's on there. Okay? I'm sorry, 185 nucleotides is the biggest one on there. All right? So they're not real large RNAs that we see here. What are the functions of these things? Okay? Well, again, we don't know all the functions, but one of the things that we do know is by looking at the sequence of U1 snRNAs, we discover that it has a region that's complementary to that 5' end. Here was that consensus sequence. And look at this. This guy has a sequence that's exactly complementary to it. We could imagine that the U1 snRNA plays a role in identifying the 5' splice site. Okay? And that's exactly what happens. When we look at the sequence of events that occur, the U1 snRNA, okay, and associated protein come in here and form a bond with the 5' end of the exon. I'm sorry, the 5' end of the intron. The U2 comes in and binds to that branch point. There's that A in the branch point. Then everybody else comes together, and we're not going to worry about the order in which this occurs. But we see that the other U proteins are involved in putting all those pieces together. In other words, remember we had to bring the A over close to the 5' end of the intron. These guys are helping to position everything else so that that reaction can occur. Okay? This shows a little bit more with respect to some of the other RNAs that are involved. Okay? So here is a sequence of the U2 sRNA. And no, you're not going to worry about these sequences, so don't worry about the sequences. But I want to point out what the U2 snRNA is doing, and it's kind of cool. Okay? The U2 snRNA is binding to that region that was pyrimidine rich that had an A. And if you look at that and you look at the consensus sequences for it, what you discover is that base pairing the U2 snRNA to the branch region causes this A to stick out like a sore thumb. This A is the one that is going to attack that phosphodiester bond at the 5' end of the intron. So this guy is sticking out of here like a sore thumb. There's no question which one is going to react. It comes and reacts at that point and makes that attack on that phosphodiester bond. Okay. Blah, 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 blah. This shows us simply that RNA polymerase can be moving along and splicing can occur as transcription is proceeding. Okay? Splicing can occur as transcription is proceeding. That's not surprising because they're both occurring in the nucleus. Now, splicing is important because it's bringing exons together. If we have problems with splicing, we're not going to make the proper RNAs that are needed by the cell. If those happen to be messenger RNAs, then we're not going to have the proper coding for the proper proteins that we want. So you might wonder, is it possible that there are diseases, genetic diseases, that arise from problems in splicing? And the answer is yes. Okay? Those problems can happen in a variety of ways. One of the ways is shown here, okay, in which a new splice site is introduced as a result of a mutation. Okay? If a splice site is introduced as a result of a mutation, we make aberrant messenger RNAs that don't make the proper coding for the protein that needs to be made. All right? If that happens, we either make the proper one over here, or we splice it incorrectly and we make an improper one over here. When we make an improper one, the cell has no use for that protein, and what will usually happen is either the messenger RNA will be degraded and or the protein that's made from it will be degraded. Well, if this is a critical protein, like something that's hemoglobin, okay, you got a severe problem. All right? So there are numerous diseases known as thalassemias that have problems associated with proper splicing. They don't make proper hemoglobins. They have severe effects in some cases on the people who have these. And these are different from sickle cell. These are usually far worse than sickle cell anemia. Okay? You may lose a complete class of hemoglobins as a result of these problems. Okay. Another thing I want to point out that's an advantage of splicing is that splicing allows for a lot of mixing and matching of exons. Mixing and matching. Okay? When people started speculating about how many proteins there were in the human genome, okay, they figured based on the studies that they had, there must be 100,000 to 150,000 proteins in the human genome. When they sequenced the human genome and discovered that there were only about 30 to 40,000 proteins in the human genome, they were quite surprised. How did this happen? We're not that much more complex than a mosquito in terms of proteins. Okay? Well, it turns out that those 30 to 40,000 proteins that we have many variants. All right? And the variants arise in different tissues from different splicing. Okay? Here's an example of a pre-messenger RNA. That is, it's unspliced. And it has some exons, or possible exons, we see located on here. This is a very simple example where we could have 1, 2, 3 exon. We have a pink exon that appears here, and we have 2 blue exons. Okay? Cells have different splicing apparatuses in different tissues. So that in some cases, like in this case shown here, in neuronal cells, one type of protein, one version of this protein can be made. In thyroid cells, a different version of the same protein can be made. Now, when I say mixing and matching, it's not like shuffling cards. Shuffling cards gets everything mixed. I would never, for example, see this guy moved up here. It's always sequential. This, 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 and then this. I wouldn't see this, it's inserted before an exon in the sequence. In this case, I can skip over something, so I go this, 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 skip this, and then get these 2. All right? But shuffling still, when I say shuffling, I'm still getting different combinations of those individual exons. Now, we'll see when I talk later about the immune system, this is tremendously important. Okay? Tremendously important. Exon shuffling allows for so many combinations of protein to be made, it's mind-boggling. And I'll tell you more about that then. Questions about this? Understand what shuffling is? Okay. Okay. And the last thing I will tell you about is that the way that introns were discovered, I shouldn't say this, not discovered, but one of the early discoveries associated with introns and how they worked was that a professor named Tom Check at the University of Colorado was studying introns. And he was studying introns in an organism called tetrahymena. The name isn't that important for us, okay? But he's studying introns in there, and he was looking specifically at, I believe it was a ribosomal RNA. And so he's interested in how this intron gets removed from this ribosomal RNA in this tetrahymena. Okay? So, like all biochemists do, he tries to take and purify the components necessary for the splicing out of the cell so he can study the reaction in a test tube. So he takes his RNA sequence that's going to be spliced, and he purifies it. He takes and he purifies a bunch of proteins that he thinks are involved in the splicing, and he mixes them together and discovers splicing occurred beautifully. Now, when this happens, you get a very big smile on your face because you think, okay, well, I've really got what I'm after. But fortunately, he didn't stop there. He's interested in understanding the mechanism by which this occurs. What can I put in? What can I leave out? What is essential for the splicing to occur? So he starts leaving out some of the proteins that he had in there, and what he discovers, that splicing still occurs. Wow, this is really a simple system. I don't need this protein, I don't need this protein, I don't need this protein. Until he got down and he removed all of the proteins. And splicing still occurred. Okay? Now when this happens, you start scratching your head. You start thinking, well, perhaps I started with something that really wasn't intact. Maybe it was spliced to start with. So I go to a laboratory and I have somebody chemically synthesize the full-length RNA for me. I put it in my reaction, and when he did that, he discovered it spliced itself. Okay? He discovered the very first example, he later won the Nobel Prize for this discovery, the first example that we know of a catalytic RNA. This RNA catalyzed a reaction on itself, okay? And the reaction involved the removal of an intron and the joining of two exons together. Okay? Now, before that time, there was not known that RNA could catalyze a reaction. It seemed that something so unusual and something so interesting should have a name, and in fact, it was given a name. He called this catalytic RNA a ribozyme. So a ribozyme, R-I-B-O-Z-Y-M-E, is a name we give to an RNA that can catalyze a reaction. Now, since then, several catalytic RNAs have been discovered, at least one of which we'll talk about later in this class, that is of extreme importance. Okay? Extreme importance. Okay, so that's a catalytic RNA. That was how it was discovered. And it's one of those examples where you just keep working and working and working, and at the end you get a result that wasn't what you expected. A lot of people would have said, I'll throw this thing out, I'm not going to do it. He persisted and was able to demonstrate that he had, in fact, something nobody had seen before. Okay, I won't go through the mechanism of that. I will tell you that there's a couple different mechanisms that are involved in self-splicing RNAs. Depends on where they come from. And it turns out that in all of biology there are two spliced RNAs that are known in the prokaryotic world. Every other spliced RNA is in eukaryotes. Well, how do we have spliced RNAs in prokaryotes? Based on what I just told you about self-splicing RNAs, you would probably think these must be self-splicing because the prokaryotes aren't going to have the proteins and so forth necessary for helping to catalyze the removal of the introns, and you would be right. The two examples that are known in prokaryotic systems, okay, are self-splicing RNAs, and they're associated with a bacteriophage. There probably may be other examples out there. We haven't studied every possible gene in the prokaryotic world, but only two that are known. Every other one is a eukaryote. Okay, that's the last about what I want to say of RNAs. Any questions or comments about that? Okay. Well, if I can keep your attention, we will have a song, but before we have the song, we have to have some background. We're turning to translation, so you might begin to guess this song is going to be having something to do about translation, and it does. Okay, now, I said when I began talking about transcription that I'm very picky that you don't mix the terms transcription, translation, and replication. So I'll repeat here, translation is a process whereby messenger RNA sequences are converted into amino acid sequences. So the sequence of nucleotides in the messenger RNA dictate the sequence of amino acids in the protein that's made. I'm sure all of you know that translation involves complexes called ribosomes. Ribosomes are something that look like this. We'll say more about this structure later, but ribosomes contain about 70 proteins. They have two components, what's called a large subunit and a small subunit. Between those subunits, a messenger RNA runs. A river runs through it. Okay? Between these two subunits, a messenger RNA. So we can see the messenger RNA here snaking through these ribosomes. There are three sites inside of this complex called E, P, and A, okay, that we'll say more about later. Okay? And what you see coming off of here is the amino acid polypeptide chain that's being made in that translation process. This guy comes snaking out of a hole at the top of the large subunit. The large subunit has a name. It's called, in prokaryotes, it's called the 50S subunit. The S refers to size. The small subunit has a name, and in prokaryotes, it's called the 30S. 30S and 50S. Okay? As I said, the messenger RNA goes between them, and the ribosome basically moves down the messenger RNA a chunk, a chunk, a chunk at a time. Each chunk corresponds to 3 nucleotides. Okay? When we are translating a sequence, we are joining 2 amino acids together to form peptide bonds. This joining is occurring, and I want to emphasize this, on the end of a tRNA. Translation is occurring with a polypeptide joined to a tRNA. Here's our first tRNA. Here's an incoming tRNA. Okay? A polypeptide bond is joined between this guy and this guy. Here's our first one up on top. Here's our second one here. The new polypeptide chain is joined to the one that has come in. We bring in another one, it'll be joined to the one that's come in. This is what we refer to as the amino end of the protein. This is the carboxyl end of the protein. Proteins are made in the amino to carboxyl direction. That corresponds to the 5' to 3' orientation of the messenger RNA. So just like we saw replication going 5' to 3', and we saw transcription going 5' to 3', we also see translation moving 5' to 3'. At the end of translation, what will happen is this bond, actually this bond right here, between the tRNA and the polypeptide chain will be broken, and the polypeptide chain will be released. Okay. So we need to think a little bit about how this overall process occurs. Okay? Where is my genetic code? Okay. Translation occurs under the direction of what's called the genetic code. Okay? The genetic code is a sequence of 3 nucleotides that specifies things for the ribosome to do. That means that the ribosome is reading the messenger RNA 3 nucleotides at a time. Each 3 nucleotides it reads tells the ribosome to do something. Okay? Well, 3 nucleotides corresponds to 64 possible mixes and matches of bases, because there's 4 times 4 times 4, that's 64. Okay? The genetic code is read in those 3s, and what we see here is actually what the genetic code is. No, you're not going to memorize it. But you can see that UGU, for example, specifies cysteine. CGU specifies arginine. Okay? There are 20 amino acids. There are 64 possible codes. That tells us that many amino acids have more than one code. 3 bases, the code is called a codon. So 3 bases of the code correspond to a codon. The amino acid methionine is almost always used to start the synthesis of a protein. We therefore refer to the sequence AUG as being a start codon. AUG is used only for methionine, and methionine is almost always used to start the coding of a protein. Yes, you should know AUG is a start codon. There are 3 codons that are what we refer to as stop codons. And what they're telling the ribosome to do is stop translating, stop making a polypeptide chain. This is the place for you to release that polypeptide chain that we saw so it can go out, it can fold, and it can do good things. Okay? The 3 stop codons you should also know: UAA, UAG, and UGA. Every other codon tells the ribosome, here's the new amino acid to build into the chain. Put this one in. That's what every other codon does. Okay? So, if the ribosome reads an AUG and then it reads a GUC, it's going to make a polypeptide that starts with methionine, linked by a peptide bond to valine. If that's followed by an ACU, linked by a polypeptide bond to a threonine. And if we have a stop codon, that would be the complete length of that. Does that make sense? Okay. So that genetic code is essential for us to make what we want. Now, one of the things I want to point out about the genetic code is if we look at these, a lot of these have common groupings, and these common groupings are interesting. Okay? Here's alanine. Alanine looks like GCU, GCC, GCA, GCG. You see a pattern there. I could write the coding for alanine as GCX, where X is any nucleotide. Okay? To make an alanine, the 3rd base doesn't even really matter very much. The first 2 are what matter. If I had a DNA that had a mutation that changed a GCU to a GCA, when it was translated, it would have no effect. Because in both cases, it would make an alanine. The 3rd base in most of the genetic code is not nearly as important as the first 2 bases. The 3rd base is what we call a wobble position. Valine, I can have a GUX. It doesn't matter what it is out here. Even for those that only have 2, let's say phenylalanine, UUUUC, notice pyrimidine, pyrimidine. UU pyrimidine takes care of it. UU purine means a leucine. Okay? So that 3rd base is not nearly as critical in the overall process as are the first 2 bases. Okay. Now, let's go back and look at tRNAs relative to that, and then we shall have a little music. Okay? You saw something like this the other day. I showed you a schematic of a transfer RNA. Transfer RNAs have a very, very important function. They carry, at one end, a sequence that is complementary to the codon in the messenger RNA. This is called the anticodon. Look at that I. Remember the inosine we talked about a while back? There's inosine. Inosine has a significance for the genetic code that I'll talk about later. I won't talk about that today. But you can see the basic structure that we saw the other day. There's our CCA sequence at the 3' end. There's that cloverleaf-like structure that we see there. There are some modified bases in here. There's pseudouridine, etc. Okay? But the most important things for our purposes is the anticodon loop and the place where the amino acid gets attached, which is at that 3' hydroxyl of the transfer RNA. The role of the transfer RNA is to carry the proper amino acid to the ribosome so it can be incorporated into the growing polypeptide chain. Okay? This shows the pairing that occurs between the anticodon on the top and the codon on the bottom. Well, duh, Kevin, I know that. All right? Well, this is one of the most commonly confused things people make on my exams. I say, what's the sequence of a codon? Where's the wobble base? The wobble base is at the 3' end of the codon. Where's the wobble base in the anticodon? It's at the 5' end. Don't confuse those two, okay? Because, remember, these guys are antiparallel. To form this base pairing, we have to have this. You'll notice that I is in the wobble position. As I said, I'll say more about that next time in terms of the importance of that. Okay, any questions about that? You guys up for one song to finish off the week? All right. This is an old song I wrote to the tune of an old Beatles tune called When I'm 64, and you'll see why. It's called the Codon Song. I hope you can help me sing it. Building of proteins, you ought to know, needs amino A's, peptide bond catalysis in ribosomes, triplet bases, 3-letter codes, mixing and matching nucleotides. Who is keeping score? Here is the lowdown. If you count codons, you'll get 64. Da 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 da. Got to line up right. 16 sRNA and shine down garnosite. We'll talk about that later. Da 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 da. You can make peptides every size with the proper code. Start codons positioned in the P site place. Initiator tRNAs. UGA stops and AUGs go. Who could ask for more? You know the lowdown count of the codons. There are 64. Da, 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 da. See you guys on Monday. You too. Take care. Take care.